Chapter 12, Section 3, Transcription and RNA Editing. RNA molecules are produced by copying part of the nucleotide sequence of DNA into a complementary sequence in RNA, a process called transcription. Transcription requires an enzyme known as RNA polymerase that is similar to DNA polymerase. During transcription, RNA polymerase binds to DNA and separates the DNA strands. RNA polymerase then uses one strand of DNA as a template from which nucleotides are assembled into a strand of RNA. The process of transcription is shown in Figure 12-14. How does RNA polymerase know where to start and stop making an RNA copy of DNA? The answer to this question begins with the observation that RNA polymerase doesn't bind to DNA just anywhere. The enzyme will bind only to regions of DNA known as promoters, which have specific bases. In effect, promoters are signals in DNA that indicate to the enzyme where to bind to make RNA. Similar signals in DNA cause transcription to stop when the new RNA molecule is completed. RNA editing. Like a writer's first draft, many RNA molecules require a bit of editing before they are ready to go into action. Remember that an RNA molecule is produced by copying DNA. Surprisingly, the DNA of eukaryotic genes contains sequences of nucleotides called introns that are not involved in coding for proteins. The DNA sequence that code for proteins are called exons because they are expressed in the synthesis of proteins. When RNA molecules are formed, both the introns and the exons are copied from the DNA. However, the introns are cut out of RNA molecules while they are still in the nucleus. The remaining exons are then spliced back together to form the final mRNA as shown in Figure 12-15. Why do cells use energy to make a larger RNA molecule and then throw parts away? That's a good question, and biologists still don't have a complete answer to it. Some RNA molecules may be cut and spliced in different ways in different tissues, making it possible for a single gene to produce several different forms of RNA. Introns and exons may also play a role in evolution. This would make it possible for very small changes in DNA sequences to have dramatic effects in gene expression.